Okay, so when we left off, we had been talking about restriction enzymes. And I kind of give you an overview of how it is possible to take a gene from a species and to put it into the DNA of another species. And that's called recombinant DNA. Genetically modified organisms are organisms that have had this happen. They have the DNA of more than one species. Um, I want to talk a little bit more about that in the process. I would like to do a lab next week, but I'm still trying to figure out how to do that. With being gone on Monday, kind of makes it harder to do because we'd have to probably do it on Wednesday, but we'll see. So as we spoke before, restriction enzymes are found in bacteria. And so they're isolated from bacteria, and their job is to cut DNA at specific locations. And for the bacteria, that serves a purpose of ensuring that any viral DNA or genetic material that gets inserted into them, there are viruses that just infect bacteria. Anything that might infect them will be broken down by these restriction enzymes. So they're kind of a defense mechanism. But we have found a way to actually use them to our benefit. They cut the DNA at DNA sequences that are called restriction sites. And they always cut at the exact same sequence. And they give you these things called restriction fragments, which are basically just the DNA cut up into smaller chunks based on where they're cutting, where they see that sequence. Wherever they see that sequence, they will cut. The most useful enzymes, restriction enzymes, are the ones that cut the DNA in a staggered way. So you end up with unpaired nitrogen bases, where one end is a little bit longer than the other of the DNA, the double strand. One strand is a little bit longer, and we call those sticky ends. And they're sticky ends because they would like to come back together with its pair nitrogen base. I'm going to show you a little video of that in just a second. Sticky ends will bond with complementary sticky ends from other fragments. So if you cut two pieces of DNA with the same restriction enzymes, they will end up with the same unpaired bases. And as a result, when you bring them back together, or when you bring them together, that will cause them to want to stick together. The only thing that really needs to, in addition to that happening, is you need to, the sugars and the phosphate won't be stuck together. So you have to have something to seal them together. And you do that with our enzyme that we've used in the past, which is DNA ligase. So DNA ligase will join the two ends of the sticky end fragments, the phosphate sugar backbone. It'll join those pieces together. So that allows researchers to join two pieces of DNA from different sources, from different living organisms, which is kind of a cool / kind of weird, great thing that we can do. All right, so I want to show you this video, but I don't want to draw on it. There we go. Huh? Why? Oh, wait, what? There's open link. There we go. Open link. I also put this in the agenda if you want to look at it again. It's short, not terribly. Really? Come on. You worked before.
Different restrictions between the bases A
So then if you cut another piece of DNA from a different organism with that same restriction enzyme, you'll get those same sticky ends. And you'll be able to take one of these pieces and then put together with that other piece of DNA. So you'll be able to, quote, insert it into the new organism. But sometimes that only works if you get these things called sticky ends. So this next one is on what happens when you don't.
restriction enzyme
And therefore they're not helpful. So some restriction enzymes from bacteria will do that. They're called blunt ends. And when that happens, they're blunt and there's no piece sticking out. And because there's no piece sticking out, they won't work. So you can't then tie two pieces from different sources together. So that won't work for that. Right. Let's see if we can get back here. So this is the next few slides are going to show that whole process. This is kind of an overview on this one, and then the next three, break it down a little bit of that slide. And I got a lot of lag going on today. I don't know why. Evidently, the computer knows it's Monday too. So here I have, remember the bacterial plasmid is that circular loop of DNA that you find in bacteria. It's not their main piece of DNA, but it's a separate piece that contains maybe 50 to 100 genes. And here we have a spot in light green where there's a restriction site. And so that's where that enzyme will be able to cut the DNA. And when it cuts that DNA, it results in these, oh, look there, it's the TTAA, just like it was in the video. Okay, so now we have these base pairs that are single. They're no longer paired together with their matching nucleotide, and they're not happy about that. They would like to come back together. They would like to stick back together. So we're going to take that piece of DNA, and now we have basically an open circle. So now instead of having a circle, we have it a little bit open, and we've got sticky ends on either side. So now what we can do is we can take that we can cut another piece of DNA with the same restriction enzyme, creating these same, and it's shown in pink here to show you that now you're talking about a different organism coming from a different source. It still has the AATT sticky ends, and now you can put it into this piece up here, and you end up with now where that loop was open, now it's closed, but that closed part, that closed little pink part, is with the DNA that came from a different organism. Okay, so now you've got DNA from two different sources. The last thing that you have to do is to then insert that. Oh, sorry, not the last thing. Let's talk about that too. But notice how if you look here, there's a little gap right here, and right here, right here, and right here. And those gaps are between the sugar of one piece and the phosphate of another. So that backbone hasn't been attached. So before you can go forward, you need to make sure that they're attached. And you do that with this DNA ligase. So that DNA ligase will seal it. So now you see there's no gaps here. And now I've got a newer, little bit bigger circle that's going to contain my gene that I'm interested in. And that could be a gene for human growth hormone, or it could be for insulin, or for a variety of different reasons. Okay, so to make sure that this actually is working, that we've actually inserted a gene from a different species into our bacteria, we can use gel electrophoresis, because now we can use those restriction enzymes, we can cut the DNA, and we should be able to see that those pieces are there. Now, that is one way to do it. And I'm going to talk about gel electrophoresis one more time in a little bit, but there's another way you can do it. Let's see if it says it here. The other way you can do it is, so let's say I'm interested in, do you remember quite a while ago I showed you the picture of the pig, and the pig had glowy snout and hooves? Okay. So oftentimes what people will do is they will not only insert a gene that they're interested in making more copies of, but they'll insert another gene that you can visually see right away. When you look at a petri dish, you don't necessarily know that there are bacteria on it that are going to be able to make insulin or human growth hormone. It would be nice to be able to just look at that dish and say, oh yeah, this worked. So if you insert another gene that is visual, that does some visual, obviously noticed thing, then you know right away that recombination worked. So one thing that's used extensively is putting in antibiotic resistance. So they will insert a gene in addition to the genes they're interested in that codes for antibiotic resistance. So you place them into a petri dish, and that petri dish will have some antibiotic on it. So what ends up happening is the only ones that will grow are the ones that are resistant to that antibiotic. And if they're resistant to the antibiotic, you can then infer that they also have the gene that you're interested in. So you can very quickly look at a dish and say, yep, this one worked. Look at this one, and it's like they're all dead, right? There's nothing on the plate. Nothing worked. Okay, so you can, anything that's there, must have the gene that you're interested in. So ampicillin or antibiotic resistance is one that they use oftentimes. They can also use a visual gene like the fluorescent. So they didn't just produce hogs that had that coloration for fun. They did it because it was also, when they inserted that gene in, they also inserted a gene that would make the hog have a larger, produce more muscle. And remember, you take a hog to the market, you get more money for it if it weighs more, if it's heavier. Okay, so but you can't just look at the little piglet and say, do you have the gene or not? You don't know that. But if you have that glowy feature to them, then you know that it must have also had the other gene that you're interested in. Now, that's an extreme example, and that I know of. I don't really know. I used to know a lot of farmers. I don't really know anymore, but I don't know of anyone who has glow-in-the-dark pigs. So, but that could be done, and obviously was done. All right, so that's kind of out of place. Let's go on to the next slide here. So sometimes this is a different technique that is not really, this is not recombinant DNA. This is something else. Sometimes you would like to make many copies of the DNA that you have. And it's funny because back in the day, 5 years ago, was this 2024? Yeah, 5 years ago, I'd talk about PCR and people couldn't care less. And now and would have no idea what it was, right? And now you say PCR, it's like, oh, I've had a PCR test. Like everybody knows about it, right? It's kind of funny. So PCR stands for polymerase chain reaction, and its job is to make a lot of copies of a specific kind of DNA. And it's a 3-step process. You heat up the DNA, which basically opens it up and makes it single-stranded. And then you cool it back down. And as you cool it back down, you add in new nucleotides that will bind to the now, quote, kind of like sticky ends. And that creates a new segment of DNA. And then you just keep redoing that over and over and over again. So you get a very large population of DNA molecules that you're interested in. And it's much more accurate than if you're talking about COVID, it's much more accurate than using the swab test the nasal test that they do and get the answer. They get the results back in 20 minutes. The PCR is much more accurate, but it takes longer. right? Because they're taking the DNA from your body and they're isolating out the DNA that's foreign, and they're producing many copies of that to see if you have the COVID DNA or not.
Wait, how does like saliva have DNA?
Every one of your, you've got, well, that's a good question. So, like on Friday and Thursday with my kids, I did a lab, my freshmen, I did a lab where they did cheek cell extraction. And basically they scraped their gums for a minute. And in doing so, in the saliva, there was skin cells, cheek cells. And those cheek cells have DNA in them.
Yeah. Okay. I feel like when you're doing the COVID test. Wait, were we like doing spit tests though? Yeah.
Yeah, you were doing a spit test, yeah.
We're not like rubbing like our cheek though. Like we're just.
Right. Well, I think they're just more sensitive. Their testing is a lot more sensitive than me with my Gatorade and alcohol test that I use, rubbing alcohol test that I use. But yeah, it's the same idea. It's funny because so on Friday the kids did it in class, and I remember one of the things they told you when you were taking those COVID spit tests was not to eat, right? So we extracted out the DNA from their cheek cells on Friday, and one kid had like a large amount of DNA in their test tube, and then right in the middle of their DNA, you know where I'm going with this, was like a little piece of Flaming Hot. It was so gross. Cheetos, yeah. Because it was after lunch and all the kids had eaten. It was like, oh, that's gross. Anyway, so one of the key things that allowed us to do this came from the fact that we don't use, if you remember, when we are making copies of our DNA, we use polymerase, right? DNA polymerase to add on new nucleotides. And the problem with that is when you heat up a DNA molecule, you break apart the hydrogen bonds between the A's and the T's and the C's and the G's, the nitrogen bases. But when you do that, if you just use a regular DNA polymerase like we find in our bodies, when you heat it up to get the DNA to split apart, you also denature the enzyme. So that's no bueno. That doesn't work so well. So by denature, I mean do what? I'm going to change the shape. So if I change the shape, it no longer works. It no longer can fit with that DNA molecule, and it can't put on the A's, C's, T's, and G's. But there's this other polymerase that they found in a bacteria, again, called Taq polymerase, and I'm going to move that around by accident. And Taq polymerase is found in bacteria that you find in hot springs. So it already lives at close to boiling temperatures. And that will not be denatured by high temperature. So you can add that polymerase in there, and then that will add on more fragments of DNA and not have a problem. So you can get large amounts of DNA. And then once you get large amounts of DNA, now you can do something with it, like a gel electrophoresis, or check and see if you have that COVID DNA. So you can take, Max, like you were saying, you can take a fairly small sample that where you're just spitting, and you can amplify out that DNA, and you can say, okay, yeah, no, they really do have a large amount of COVID, so they are COVID positive. Okay. What do we got? 6 minutes? I think I got this. So I did want to talk about the lab you did on, I don't know when, a while ago, right? The gel electrophoresis lab, forever ago. I have been having problems as of late getting results on that. And that's why I didn't show it to you right away. There was nothing to show you. So I wanted to talk a little bit about it because now is the time that this kind of all comes together. So in our lab, we started out with the scenario that we knew that Valerie had breast cancer. Okay. She had the P53 gene. What was the P53 gene? What is P53? It is a tumor suppressor gene. Its job is to, if a cell looks like it's messed up, P53 will generate a protein that will pull aside that cell and say, nope, you're not ready for prime time. There's something wrong with your DNA. We won't let you go forward into the next part of the cell cycle until you fix it. So that's P53's job. If the P53 gene is mutated, that won't happen. There's nothing to check the DNA, and cell growth, cell division can continue unchecked. So the reason we did this particular lab was to examine not only, we knew she had breast cancer. So if you take a biopsy, the cells from her biopsy should show that she has 2 mutated copies of the P53 gene. You need at least 1 copy of the P53 gene functioning in order for the cell cycle to be stopped if there's a problem. Okay. So we knew that she had breast cancer. We knew that there would be 2 mutated copies of the P53 gene in one of her, in that biopsy. Now, let me talk about the other lanes that we had. Go away. So there was lane 1 was just standard DNA fragments. These are fragments that we already know how big they are and how far they're going to travel in the gel. So like one is 10 base pairs, another one's 200 base pairs, that kind of thing. So you can kind of get an idea. It acts as a ruler to measure everything else against. Then there was also control DNA that was in lane B. And this was just some normal P53. And remember, you get 2 P53 genes. Where do you get them from? Where do you get one of your genes from? Your mom, and you get the other one from your father. Okay. So you would have, in this case, control DNA, normal P53. Now, what do I mean by normal P53? I mean that if I take a restriction enzyme and I cut the DNA, it will cut a fragment of a certain length. Okay. If there's a mutation, that P53 gene will get cut in a different location and it will be a different size. Okay. It won't be the same size if it's mutated. Okay. And I'm going to show you the results in a second. So Valerie's DNA. This came from her blood. One of the questions that the lab posed was, okay, we know she's got breast cancer, and this is actual things that they would do if there was a scenario like this. They would take cells from the bloodstream to see if the cancer had spread to other parts of her body. So by checking the bloodstream, you can see if it's metastasized, and if it's metastasized, it's spread, and that creates a whole another set of problems that we have to deal with in terms of treatment. Lane D was from Valerie's actual tumor. This was the biopsy. So this one should show a different size chunk than we should see from her E. What we're hoping is her normal breast tissue. So they took tissue from the tumor, and then they took tissue from beside that tumor to see if that had spread to a different location. All right, questions? All right, so sad face. I don't know why this doesn't work for me. Yeah, it is. It's a sad face. See here? It says right there. It's a sad face. Yeah, sad face. All right, let's clear the screen. Okay, so A is our standard ladder. That's our ruler. And these segments here, let's just do red because it's a good color, would always show up. B, what do I say B was? Normal P53 gene? Okay, so I cut it with the restriction enzyme, and this is another control. This is what it should look like if you have normal P53. If you are homozygous dominant, what does that mean?
Same.
Yep. Big. And so I'm going to say P, big P, meaning normal P53 gene. So that's what I have for here. C, I believe, was her, was it her bloodstream? Yeah. Okay, bloodstream. Good, thank you. So C was her bloodstream. Notice how there's a segment here, just like there is here. So that's telling us that she has one good P53 gene, because one of her genes went to the same location as the control lane B did. But she did have one copy that was mutated, and we already knew that, right? We already knew she's going to have one copy that's mutated. So what's her genotype for that tissue? She's heterozygous. Good. She's big P, small p, because those two pieces represent where the restriction enzyme cut the fragment in half. So it's smaller because instead of just being one fragment, it cut it right in the middle, or not right in the middle, but it cut it in a way that gave you two different sized fragments, two bigger or smaller fragments if these are down here. Smaller. They're going to be smaller because they're traveling faster. Okay. And then D was, what was D? To tumor. Uh-oh. What is she here?
Oh, she's saying this in her blood.
Yeah. So she is, we knew that already. We already knew that she had breast cancer. So though she has two bad copies of the P53 gene there. Okay. And then was the last one, what was the last one?
Other breast tissue.
The other breast tissue. So in her regular or other breast, also a good copy and a mutated copy. So what is the conclusion from this? Has it spread? No, it is not spread, because in her blood, there's still a big P. And in her other breast tissue, there's still a big P. As long as she's heterozygous, as long as she has one good copy, it is not spread. It is not mutated. It is not causing cancer. Okay? That's it.